Hello, and welcome to this masterclass on how to analyze your Oxford Nanopore sequencing data. My name is Brian Catano, and I'm a Senior Product Support Scientist at Oxford Nanopore Technologies. This masterclass series is designed to guide you through the entirety of the Oxford Nanopore sequencing journey, from sample to answer. In this masterclass, we will focus on the analyze component of this workflow, discussing everything from setting up your sequencing run to analyzing your data. First, we will begin with an introduction to how raw nanopore sequencing data is produced and how the process of base calling works. We'll then look at how to use the software MinKNOW from setting up your sequencing run to calling canonical and modified bases to performing real-time targeted sequencing. Next, we'll talk about what you can do with the intuitive Epitome data analysis platform and the wide range of Epitome workflows available for analysis of your Oxford Nanopore sequencing data. Learning bioinformatics for biologists or anyone inexperienced in programming can entail a steep learning curve. The scientist generating the data might need to pass it on to the bioinformatics department for data analysis, therefore losing ownership of their data and possibly missing the full understanding of the workflow. With our platform, you can have full ownership of your data and choose the best solution that suits you, from user-friendly point-and-click software to the underlying command line tools. Oxford Nanopore's goal is to enable the analysis of anything by anyone, anywhere, and this extends to data analysis as well. We offer user-friendly analysis solutions that are accessible to all levels of expertise, scalable to all sorts of hardware, and versatile to all sorts of analysis needs. You can choose where to run your analysis depending on your needs and infrastructure, in the cloud, on your desktop, on the sequencing device, or on a server. At Oxford Nanopore, we provide not only sequencing solutions, but also a wide range of options for data analysis, from basecalling to QC to a variety of downstream analysis and reporting tools. Here, you can see a list of software and which stages of the analysis they cover, what level of expertise is required to run these, and which kind of support we provide. As well as the software controlling the sequencing, we have other software tools providing full bioinformatics workflows for specific genomic analyses. You can choose our user-friendly interface to run them in the cloud or locally, and command line options are also available. Today, we'll be focusing on our local and cloud-based solutions, but for information on more advanced usage through the command line, you can visit our website. Those with more advanced bioinformatics expertise can also choose to use research tools and tools developed by members in the Nanopore community. To get started, I'd first like to introduce the base calling process and some common file formats. Generally, a data analysis workflow will start with the raw data and end at an answer to your biological questions. But it's important to remember that this is not a one-way street. Data requirements at each stage will place demands on each previous stage, so be sure to consider what the final data requirements will be before you begin your experiment. As we work our way through this workflow, pay special attention to the file formats being used at each stage, as bioinformatics is really all about converting data from one file format to another that is more suitable for addressing your biological questions. For example, you may want to align your nucleotide sequence in FASTQ format to a genomic reference, which will yield alignment files in the SAM or BAM format. From these alignments, you can call variants and output that to a standard variant call file or VCF format. I will point out common file formats of interest as we move along. As a recap, Oxford Nanopore offers a highly scalable method for sequencing DNA or RNA. After a sequencing library is added to the flow cell, DNA or RNA molecules translocate through the nanopores. Independent channels measure how the current passing through a nanopore is disrupted by this process, which gives us an electrical signal that serves as our starting point. This signal is the raw data that gets base called to a nucleotide sequence. Basecalling is a process of converting that raw electrical signal generated at the nanopore into a nucleotide sequence using state-of-the-art algorithms. The input to basecalling is the raw signal stored in our POD5 file format, while the output is the nucleotide sequence in standard FASTQ or BAM format. Here's some more information on file types. The standard FASTQ file type is used for storage of basecall data, and it's usable in common downstream analysis tools. This is a text-based file format that has your nucleotide sequences in A's, T's, C's, G's, or U's, and it being a text file, it's very amenable to compression, but it's already relatively small in size. If there's no desire to rebase call your data or to use raw signal data for any complex tertiary analysis, then FASTQ alone is generally sufficient. If you wish to keep the raw data from your sequencing, this is contained within the POD5 file, which is based on the Apache Arrow file format. This raw data enables you to rebase call the original read without needing to resequence, and it can be used in some analysis approaches, for example, around calling modified bases, or for training custom models if required. Due to the presence of the raw signal data, this file format is considerably larger than the FASTQ. One final type to consider is the BAM file format, which can be employed if you choose to carry out modified base calling, or when aligning your reads to a reference. These can be converted back to FASTQ if needed, but they generally work as the input for downstream analysis tools as well. The total volume of storage required for this will vary by application. Now, let's look at the analysis that's available out of the box when you start a sequencing experiment in MinKNOW. MinKNOW is the main software that manages your device. It controls your sequencing runs. It performs analyses at different stages, from base calling, including methylation calls, to further data analysis, including quality control steps. And there are also additional features, such as adaptive sampling, which enriches for targets of interest while sequencing. MinKNOW also generates a report at the end of the run automatically. As well as controlling the sequencing experiment and saving your raw data to disk, MinKNOW is also capable of base calling your reads live during the run, or after the run is complete. For advanced users wanting to take advantage of the latest state-of-the-art features and updates, the standalone base caller Dorado can be run at the command line and is available on our GitHub. You can choose between 3 different accuracy models given your computational resources and your time to answer requirements. There is a fast model that requires less computational power and less time to result, and this is perfect when turnaround time is an important consideration for your application, or when sequencing in the field, to give a couple of examples. The super accuracy or SUP model gives the best accuracy, but it requires a lot more GPU power to base call. The high accuracy or hack model is a great middle ground between these two options, so it's a really nice balance between speed and accuracy. For base calling, we recommend that you use the most accurate base call model as possible, given your computational setup and your time to answer requirements. In our end-to-end workflows, we will provide guidance on which base calling model to use. Thanks to the ability to sequence native DNA and RNA with Oxford Nanopore, it's also possible to directly detect base modifications in your data. You can enable methylation calling as part of live base calling seamlessly in MinKNOW. The tool that enables methylation detection, Remora, runs in parallel to the base caller during live sequencing and base calling. Remora is also available as a standalone tool on our GitHub page, accessible via the QR code here. When you start sequencing, MinKNOW will start producing a number of graphs, which you can use to help assess the quality of your sequencing run using a very intuitive interface. As you can see here, this includes information on channel states, your total output, the quality scores for your reads, and your read length distribution. Runs can also be monitored from the device as you're sequencing, or remotely from a phone or laptop on the same network through the MinKNOW Connection Manager. At the end of your experiment, MinKNOW automatically generates an HTML report for each run. MinKNOW also has a few other real-time or post-run analysis capabilities, such as barcode demultiplexing, alignment, enrichment, or depletion using adaptive sampling. Adaptive sampling is a really interesting technique that's unique to Oxford Nanopore. This is a PCR-free, real-time, bioinformatics-based method for targeted sequencing that enriches the data while still conserving epigenetic information. You can perform this without any special library prep steps, such as bisulfite conversion. It's all done in silico during sequencing itself. How this works is, while a DNA strand is passing through a nanopore, MinKNOW can very quickly, in real time, base call and align the very first part of that read to a reference, and then make decisions based on whether or not it matches your target region. So, based on how you configure the store and setup, MinKNOW can either accept the read and keep sequencing it, or reject the read and kick it out of the nanopore so that the pore becomes immediately available to start sequencing another molecule that is hopefully within your target region. So this can be run in enrichment mode, where you're only keeping the strands that align to your regions of interest, or in depletion mode, such as, for instance, host depletion, where you kick out any strands that align to the reference that you provide that you don't want in your final data set. Now we will switch over to a short demonstration of starting a sequencing run in MinKNOW and see some of the initial analysis performed out of the box.
This is the main MinKNOW landing page, where you can connect to your sequencing device. Depending on which sequencing device you have, you may see one position available, or you might have multiple independently addressable positions. To start a sequencing run, we'll click the Start button in the upper left-hand corner, and this is where you can start your sequencing runs and analysis workflows, as well as perform the hardware check and the flow cell check. To get started, we'll click the green Start Sequencing button, and that will take us to the Positions page. If you have multiple positions available, they are independently addressable, so you can select one of them, or multiple, or all. As we only have one position available with this Mark 1D device, that will auto-select for you. To get started, we'll need to set an experiment name. We'll call this Masterclass Demo. And optionally, you can set a sample ID. I'll call this Demo 1. The flow cell ID and the flow cell type should populate automatically, but if you see anything that's wrong here, you can manually enter your flow cell ID or manually change the flow cell type. Next, we'll click Continue, and this will take us to the Kits page. On this page, you can select the sequencing kit used to prepare your library. There are some filters up here where you can select whether you're using an RNA kit or a DNA kit, whether you're using a PCR-based or PCR-free method, and if you're multiplexing. You also have a control option. When you're sequencing a lambda phage control, you can select this and it will automatically default to a 6-hour runtime. Depending on which kit you select, you may have optional expansion packs, such as if you're barcoding your library with the PCR barcoding expansion pack 1 of 12, and then feeding that into the ligation sequencing kit. You can select that here as well. We'll go ahead and select the native barcoded sequencing kit 24 and click Continue. You'll next be shown the Run Configuration page to review your selections, and this is all you need to start a sequencing run with the default settings. We choose very sensible defaults to work in a broad range of applications for as many people as possible. So you can go ahead and click Start and then immediately start sequencing and answering your biological questions. For now, I'll scroll through and show you the various options available to configure. At the top, you'll see the experiment name and sample ID, as well as the flow cell type and sequencing kit being used for this experiment. In the sequencing and analysis section, here's where you can turn on basecalling and modified basecalling, and where you can select either the fast, high accuracy, or super accurate model. You can also enable or disable barcoding, barcode demultiplexing, as well as edit some options if you want to trim your barcodes or require both barcodes on both ends, as an example. You can load a reference file for aligning your reads automatically to that reference during the run and outputting that to a BAM format. You can also start an adaptive sampling experiment, either in enrichment or depletion mode, as we discussed previously. And again, you load your reference and your target coordinates from a bed file to get started. In the data target section, you can select your run length as well as your flow cell data targets. So I can change this from the default of 72 hours to any other runtime I want, as well as setting it to the flow cell's end of life. So if you've generated a lot of data and there are no more pores left for sequencing, you can have the run stop automatically instead of running it until the time that you've manually set. You can also set a flow cell data target, either by base call bases or estimated bases. So for example, if you know you only need 10 gigabases of data to answer your biological question, you can set this to 10, and MinKNOW will automatically stop the sequencing run once you reach that threshold, so you can wash and reuse your flow cell for a later experiment. I'm going to click cancel and just continue with the defaults here. In the output section, you can change your output location, and you can also change your output formats. So you can choose whether you want FASTQ files or BAM files or both. You can also change your output frequency either by number of reads per file or by having a file output at certain time intervals, 1 hour, 10 minutes, hourly. If you'd rather have one large file containing all of the reads at the end of the run, we can click end of run, and when the run finishes and all your data are base called and processed, it will concatenate all the files together into one big file. You can also enable filtering here, either by Q-score or read length. This is what sets your pass and fail filtering. It's determined by base call data. So if you have live base calling on, after it base calls, MinKNOW will check the Q-score of the read and, based on the threshold you set, bin it into a pass or a fail folder. You can also set minimum and maximum read lengths for filtering. Another interesting thing when you're running the same samples over and over again with the same parameters is that you can save your configuration to a template. And then for all your subsequent experiments, you can load that configuration so you don't have to manually adjust your settings every time. You can have that done consistently by just loading this up from a template. To get started with sequencing, we'll just click the green Start Sequencing button. And we'll see that this position will now be changing states. It will take a few minutes to get to temperature and perform a few checks to make sure all the pores are good, make sure all the membranes are good, make sure the voltage and the temperature are correct and that they are optimal for sequencing. So we'll step away here and check back in about 15 minutes after the run has started. So here we are a few minutes after the run has started, and we can see our position is in a sequencing state. So let's check in on the run's performance. We'll just click here to navigate through to the experiment. On the first page, we'll see details on the settings that we've configured, the experiment name, the sample ID, current output directory, total runtime, things such as read counts, how many bases are estimated to have been sequenced and base called, and more. Clicking to the right to the next plot, and we'll see the channel states panel. This is a dynamic view of what each sequencing channel is doing on the flow cell. So we see here we are somewhere around 410 to 420 active channels sequencing at any given time. So light green are the channels with DNA actively translocating through the nanopore to generate signal data that will base call to get reads. Green is good. When you start your experiment, you want to see a sea of green here. When sequencing has progressed for about an hour, you'll see a number in the upper left-hand corner called pore occupancy. This is essentially what percentage of active pores have DNA translocating through them at any time. Ideally, your pore occupancy is above 90% after the first hour for the majority of sequencing experiments to get you as much data as possible. You also have a cumulative output plot where you can see how many total reads you have, as well as how many have passed and failed, depending on the filtering options that you've configured when starting your run. In this case, we only set a Q-score filter of 9, so this is how many reads have passed that filter. We can look at this in terms of number of reads or number of bases, because fragment lengths are a variable in the library. So 2 different reads of different lengths will count the same in terms of number of reads, even though the longer read will have more bases represented within it than one of the shorter reads. We also have a read length histogram where you can look at this in terms of read length or counts, as well as estimated or base called bases. And there's a slider here that you can use to drag to zoom in on a particular area of interest. So for example, if we want to look at reads between 0 and 11 kb, I can slide this over and look at just those reads. And you can reset the zoom selection with this reset zoom selection button. About 15 minutes into the run, we see we have an estimated read length N50 of about 8.5 kb, and this will update over time as more reads are generated. We also have a Q-score histogram plot separated by colors for passed and failed reads, and we can see here we have a nice big peak centered around a Q-score of about 18 or 19. It will give you a summary and show you the filter criteria here on the same page to give you context to your histogram. There's also barcode hits. So if you're performing multiplex sequence, then you are demultiplexing live during the run. You'll be able to see which barcodes have been sequenced and in what amount live during the run. As before, we can look at this in terms of reads or bases. We can sort by barcode number, or we can sort by the amount that you have in either direction. And these are all the default plots. If you want, you can adjust the plots here using the configuration button to access more or less information as you wish. Generally, these defaults are what you need to make decisions on your run, such as when to pause, wash the flow cell, reload with more library, etc. But we'll select all and have a quick look through the rest. The same channel states panel is available, but now we have a pore activity plot. This is the same as a channel states panel, except charted over time. So you can see roughly the proportions that all the channels, the states that all the channels were in as time progresses over your experiment. We have a pore scan results plot. By default, every hour and a half into a sequencing run, MinKNOW will temporarily pause sequencing to do a global assessment of all the channels and see how many pores remain available for sequencing on that flow cell before restarting to make sure it's selecting the best channels to give you as much data as possible. So you can track your rate of pore loss over time as these mux scans come in. Cumulative output plot as before. The read length histogram plot. You also have a translocation speed plot So this shows you over time the speed at which DNA or RNA is being actively translocated through the pore. This should remain stable over time if you're using our latest sequencing chemistries. The Q-score histogram plot remains unchanged, but we also see a Q-score over time plot. This will give you a time course representation of the quality scores of your reads as time goes on and more data are generated. The barcode hits plot. There's also a temperature history plot, so you can track and ensure that temperature is remaining stable over the course of the run. If anything deviates, MinKNOW will give you a warning, but generally you should see this remain stable with all of our latest devices that have integrated temperature control. There's a bias voltage history plot, so you can see the voltage changes that are occurring depending on the different stages of the sequencing script, whether a pore scan is occurring, whether a MUX scan is occurring. And as sequencing chemistry gets depleted, the voltage will be adjusted to compensate for that and ensure you're getting the highest quality data over time. We have a disk write performance plot where you can ensure that your reads are being saved sufficiently fast to disk. And we have a trace viewer that allows you to, for any channel here, look directly at the raw signal being measured at that channel. So here in channel 10, we can see we have some DNA actively being translocated through the pore. We are generating this raw signal. This raw signal is being base called by MinKNOW to give us a nucleotide sequence. As DNA exits the pore, you'll see the current changes. As another DNA strand enters the pore, the current will change again, and you'll see this strand continuing to sequence, this new strand sequencing to be base called and give you more reads. I'll leave you to explore these additional plots on your own and to share your feedback with the Nanopore community. We can also resize to look at just the plots in full screen here, or we can minimize to access the start, stop, and pause buttons at the top here. To navigate back to this page, you can click on the experiments page, and then in that tab select the active experiment. Or you can click on the sequencing overview tab and click on the position with the active experiment.
Now that we've talked about MinKNOW and the earlier stages of data analysis, I'd like to introduce you to Epitome. Epitome is Oxford Nanopore's fully supported desktop application for nanopore data analysis. The intuitive desktop application allows you to run a range of pre-configured data analysis workflows, which provide interactive reports and standard output files. Epitome can be run locally or in the cloud. Some workflows can be run in real time, allowing you to analyze your data as it is being sequenced. And the same workflows are accessible through both the graphical user interface and via the command line, fostering collaboration between scientists and bioinformaticians. You can also easily integrate Epitome with your own workflows, as well as tertiary analysis software. So how does Epitome work? Your DNA or RNA is sequenced on your sequencing device, and as we've seen, this is all managed by the software MinKNOW. Your device will then write files to a location, and in some cases, these are then immediately available for analysis in real time, or they are available post-run. Epitome can then run in the cloud or locally, and will generate intuitive reports as well as workflow-specific results files, which can be used downstream. Epitome functions as a standalone desktop application, where you can select your desired workflow, point to the folder with your data, and then you can configure your relevant parameters for that specific workflow and start the run. Epitome comes pre-packaged with over 15 open source workflows using the bioinformatics framework known as Nextflow. The great thing about Epitome is that no bioinformatics experience is needed, and minimal software installation is required. To install Epitome as a desktop application, all you need is Docker and a relevant version of Java. Epitome features a selection of pre-configured workflows for a wide range of applications. Let's take a look at some of these now. Unfortunately, we don't have the time to cover every workflow that's available, so for more information, do check out the resources at the end of this masterclass. Firstly, we will look at base calling and then aligning to a reference. So as we've seen, base calling can be performed on your sequencing device using MinKNOW, but it can also be done using Epitome. This workflow allows you to provide base call data and then align to a reference if necessary. The workflow outputs base called sequence data that can then be used downstream in any other Epitome workflow. After you've base called your data, you can use our flexible standalone alignment workflow to align it to a reference of your choosing. The workflow uses Minimap2, which is a versatile sequencing alignment program that will align the input reads to the reference provided. The workflow will output the fully aligned reads in a BAM file format, as well as a QC report summarizing the results. If you're performing PCR-based targeted sequencing, you can use the Amplicon Epitome workflow. The Amplicon workflow can be used for the analysis of any kind of amplicons from barcoded libraries. The workflow can be run in two modes, de novo assembly mode, based upon the barcode, or variant calling mode against a reference, using Minimap2 to align the reads, and then our tool Medaka to call variants. And the workflow generates an intuitive HTML report and a BAM file containing the aligned reads. When running in variant calling mode, it will also generate a VCF file with the variants, among other outputs. If you're looking to analyze microbial sequencing data, Epitome has multiple workflows for this purpose. The workflow clone validation can be used to take advantage of long Oxford Nanopore reads to quickly assemble and validate your clone constructs by barcode. This workflow will perform a de novo assembly of all the plasmids. It'll annotate the full assembly and provide an overall quality score of the plasmid. If you have inserts in your plasmid, these will be located using the inputted primers. As an output, you can obtain an intuitive report, a consensus FASTA file with annotations for each barcoded sample, and a status table labeling a pass or fail for each barcoded sample. For information on the end-to-end plasmid sequencing workflow with Oxford Nanopore, make sure you catch the masterclass How to Sequence Full-Length Plasmids in Your Own Lab. The bacterial genomes workflow performs assembly or alignment of bacterial genomes and annotation of no references included. So various standard tools are used with Epitome doing the work for you. The assembly is performed using Fly and then polished with Medaka. If a reference is provided, alignment will be performed with Mini-Align, and then variants will be called with Medaka. This workflow offers a few optional extras. For example, it can run Proca to annotate the resulting consensus sequence, or it can use a software called ResFinder to check your sample against a database of antimicrobial resistance genes. The outputs can also include VCF files of detected variants, as well as an HTML report. If you're performing metagenomic or 16S sequencing, there are dedicated Epitome workflows for these techniques too. The metagenomics workflow enables identification of the origin of single reads from both amplicon-targeted and shotgun metagenomics sequencing. It provides intuitive reports with powerful and interactive graphics. The workflow can be run both in real time with a quick approach using Kraken 2, or post-run with a finer, more computationally intense approach using Minimap 2. By default, the workflow will use the NCBI 16S 18S RNA database, which will be downloaded at the start of the analysis. However, you can also use custom metagenomic databases. The 16S workflow is based on the metagenomics workflow, but it's configured for the database's parameters and example data that have been optimized for analysis of the 16S gene. If you're studying infectious disease, then Epitome also has multiple workflows for this. We provide end-to-end protocols, including data analysis workflows in Epitome for characterization of a range of pathogens, and you can see several of these here. The flu workflow, for example, has been designed to identify influenza A and B types. The workflow aligns the amplicon reads to a reference using Minimap2. It calls variants with Medaka, and it uses abricate to type the sample. It will then return an intuitive report with clear assignment and characterization. If you're looking for SARS-CoV-2 analysis, the workflow Arctic can be used. This workflow generates shareable reports with key information, including clade and strain identification. As well as its use in the microbial isolate sequencing protocol, the bacterial genomes epitome workflow can be used to characterize Salmonella as part of our rapid, direct from colony Salmonella serotyping protocol. There are also epitome workflows for the study of Mpox and Mycobacterium tuberculosis. If you're studying human genomics and transcriptomics, epitome has dedicated workflows for this. The human variation epitome workflow offers comprehensive analysis of the human genome in a single workflow for the analysis of SNVs, SVs, and methylation, as well as copy number variants and short tandem repeats, all from a single Oxford Nanopore dataset. As we covered earlier, all you need to do is switch on modified basecalling in MinKNOW to make sure that you record 5MC and 5HMC methylation in CpG context, as well as the canonical bases in the output BAM file. The data can then be aligned to the human genome either in real time in MinKNOW or in epitome as part of the human variation workflow. The workflow outputs annotation files and reports for small variants, structural variants, copy number variants, and short tandem repeats, as well as SNV phasing blocks. Visualizations of the aligned reads and different tracks can then be viewed within epitome, or you can open the output files in your preferred genome browser, such as IGV. For cancer research, there's also an epitome workflow available for the analysis of tumor normal sequencing data. The somatic variation workflow takes the BAM file produced by Basecalling and aligns it to the provided reference genome. It then calls SNVs and small indels between the tumor normal or tumor-only samples using CLAIRE-S, which is a somatic variant caller designed for long reads. Variants greater than 50 bases in length can be detected using the structural variant caller nanomon-SV. Analysis of 5MC and 5HMC methylation is then performed, including differential methylation analysis when using paired samples. All of this information is then generated into an HTML report, from which you can visualize the results. If you're looking at analyzing transcriptomes at the bulk or single cell level, Epitome also offers two workflows for this. If you're looking at bulk transcriptomes, whether you're sequencing cDNA or native RNA libraries, Epitome offers a specific workflow for this. After sequencing and base calling your libraries, you can run the transcriptomics workflow in Epitome through the graphical interface. To do this, you will only need a FASTQ file and a reference genome or transcriptome file. Optionally, you can perform differential expression analysis using a reference annotation GFF file. Or you can also detect gene fusions using a JAFL reference. The workflow will align the reads, assemble and annotate the transcripts, and then optionally find gene fusions and perform differential expression analysis, all in one HTML report. Epitome also offers a dedicated single-cell workflow that can be used to investigate single-cell data produced by nanopore sequencing of 10x genomics libraries. After sequencing and base calling, your libraries will be tagged with barcodes if you're looking at the cell level, or UMIs if you're looking at the transcript level. You can then run the single-cell workflow in Epitome, and from an input of the FASTQ files, reference genome, and the annotations, you will get an aligned BAM file with tags looking at the barcodes or the UMIs or the annotations, plus gene or cell expression and cell or transcript expression matrices, as well as a UMAP plot, all within one simple report. Before we wrap up, let me leave you with some additional resources. If you want to learn more about Oxford Nanopore data analysis, we provide a variety of tools and guides to quickly get you started. We recommend visiting our website to access training materials, documentation, and more, including detailed information on epitome. I'll leave the slide up here for a second so you can access these via the QR code of interest, and you can always refer back to the recording of this presentation at a later point to get direct links to all of these locations. I hope you found this useful. Thank you for watching and enjoy the rest of the Masterclass series.